Where am I? Why do I just disappear? Right. Mm, 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 mm. Ach, man. Come on, come on. Come on. Go that way. So, no, okay, good afternoon, everybody. And today we're going to carry on our discussion about membranes, membrane structure, and membrane function. Again, please remember, if you have any questions, just unmute and I'll be able to hear you. But when you finish your question, please mute again. All right, so we've said all the way through that one of the really important functions of the membrane is to allow conditions on one side to be different to conditions on the other side. So today we're going to just be discussing how exactly that is achieved. The membrane itself, the foundational structure, that phospholipid bilayer, is in itself a good barrier. It's a very good barrier. It will allow some things to get across quite easily, gases, for example, or small molecules may be able to get across. Even water can slowly get across, but very inefficiently. So it's actually a very good barrier. But the thing is, the cell couldn't possibly function just with that barrier, because the cell has to control what is on going in, coming in across the membrane, and what goes out across it. And not only at the cell membrane, but if we're talking about eukaryotes, the membranes of all the organelles themselves must be, the passage of molecules to and fro must be carefully controlled. It is the responsibility of proteins embedded in that phospholipid bilayer to actually govern the passage of molecules to and from across the membrane. And it's actually those proteins that confer a living nature to the membrane. The foundational phospholipid structure basically does not do anything. It is the proteins which are governing what moves back and forth in both directions. So we need to be able to understand some basic principles about how molecules move, what drives them anyway, in order to really understand what these proteins are doing. So I'm sure that some of this is familiar to you, probably from physics or chemistry. But we need to understand what this principle is of diffusion. And diffusion is the movement of particles of a substance so that they spread out evenly in the available space. That spreading is driven by their kinetic energy.
Hi, good afternoon. I just got here. What are we talking about?
You can just turn your mute off and listen, and then you'll hear what we are talking about.
Oh, okay.
So diffusion is the movement of particles of a substance so that they spread out evenly into the available space. It's driven by their kinetic energy. And the net movement, although each particle moves random, they're smacking into one another the whole time. And the net movement of the substance is to distribute the substance from a high concentration to low concentration. We say that that is in the direction of the concentration gradient. The substance will move from an area of high concentration to an area of low concentration. And then eventually, when everything is evenly distributed, we say that we have reached equilibrium, but this has a very special meaning. So let me show you here. First of all, this is just a simple conceptual experiment. We have some sort of barrier, not a profound barrier, but a barrier that will allow the substance to move. On one side of the barrier, we have lots of the molecules of the solute. It could be anything, a dye, it could be a salt, it could be ions, whatever. On the other side, we have very few. This is an aqueous solution. The diffusion of the substance is going to be from an area of high concentration to one of low concentration. We've put here a membrane, but a membrane which is allowing free passage of the substance. Is going to move across in response to the diffusion grade, to the concentration gradient, high to low. Eventually, it will be equally distributed throughout. This is the situation of equilibrium, but it doesn't mean to say that movement stops. It does not. Movement still carries on, but at equilibrium, for every molecule that comes across this way, another molecule will go that way. That's the equilibrium. Now the concentrations are the same on both sides. That is the drive, is to have the concentrations equal within the available space. But I want you to think about something. When this is an aqueous solution, have a look at the situation here. Okay, we've got something there, right? Dissolved in the water. Where is there less water? There's less water on this side, right, than there is on that side, because these molecules here are occupying space. Well, if there's more water on this, there's a higher concentration of water on this side than there is on that side. And whenever we think about these aqueous diffusive situations, we need to remember that water is also moving. Water is moving to try and equalize its concentration as well. Water will be moving this direction at the same time as the solvent is moving this direction. Solute, rather, is moving that direction. That diffusion of water, yes, go ahead.
Sorry to interrupt. Is there a difference between equilibrium and dynamic equilibrium?
No, it's the same. Yep. There's a dynamic equilibrium because every motion this way is compensated by a motion that way. So it's just equilibrium. That is dynamic equilibrium because there's a dynamism involved. That's all. Okay, so that's one solute, but remember, all the way through this, be thinking what's happening with the water, because that's going to become very important. So here, just an illustration that this principle applies individually to solutes, right? Here we have a situation where we have a high concentration in this solute, this side of the membrane, a high concentration that side. They still try and even out. This purple one is going to even out and have its own equilibrium. The orange one is going to even out and have its own equilibrium. They operate independently of one another. Okay, so I said to you, always be thinking about the water. What is happening with the water? The water obeys the same principle. The water also moves from a region of high concentration, of high water concentration, to an area of low concentration. The area of low concentration of water is that area which has the higher concentration of solute. If you've got a solution with lots of salt in it, it has less water in it than a solution with a lower concentration of salts. And the movement of the diffusion of water molecules, it has a special term. It is called osmosis. And we're going to watch now as water molecules diffuse across the membrane from an area of one solute concentration to an area of another solute concentration. And we do it with a simple experiment. We got here a tube, and across this here, there is a membrane. But this is now not an open membrane like the ones we were discussing earlier. This is a semi-permeable membrane. In other words, it will allow the passage of water, but it won't allow the passage of the solute. The solute cannot move to move down its diffusion gradient, but water can. So look at the situation that we have here. They've dissolved sugar in the water. Initially, we're starting off with equal volumes here, but there's less sugar on this side than there is on that side. The sugar can't diffuse this way. The sugar can't diffuse that way because this is a semi-permeable membrane. The importance of that is that our biological membranes are semi-permeable membranes. So we're looking now this is a much more realistic, much closer to the living membrane's function. It is semi-permeable. Semi-permeable because it allows water to diffuse easily. And it does not allow the solutes to diffuse easily. That's how the cell membrane works, really. It works to keep in the solutes that it needs inside the cytoplasm, and water can pass freely across the membrane. So now let's work out what's happening. The solute cannot move to equalize the concentration, but the water is driven as well. The water is driven by its own diffusive properties. It is going by osmosis. The water is going to attempt to equalize the concentration of water on that side and that side. And what is going to happen is water is going to pass across the semi-permeable membrane. It's going to dilute solute on this side, and it's going to concentrate solute on that side until the concentrations of both solute and water are the same on both sides. Okay? Are you all right with that? I'm expecting to see a microphone light up. Are you okay with that? Somebody
Now
else?
we can talk.
Huh? Are you okay, Dylan?
Yes.
Are you okay with that?
Yes,
Yeah.
Professor.
Okay, fine.
I'm good with that.
Anybody, if anybody's confused or anything, stop me and ask me, okay? Because this is an important principle, and it's not necessarily something instinctive. This looks crazy, because in fact, look what's happened. We've raised the water level. But if we look at the concentration of water on that side and that side, it's the same. If we look at the concentration of solutes, it's the same there and there. And that's a property of the semi-permeable membrane. It's a very important property of biological membranes, that they are semi-permeable. They are semi-permeable for an important reason that I'll get to later. Remember what I said right at the beginning. The living membrane, the living cell membrane, water does not pass across it very easily. It can go a little bit, but not efficiently. But in the living membrane, water passes across very easily because we have special proteins there that allow it to go. And we'll see those in a minute. Okay, this here leads to a very potentially dangerous situation for the living cell, because this passage of water is inexorable. As long as one side of the semi-permeable membrane is more concentrated in solutes than the other side, water is going to flood across the membrane to try and dilute it. This is dangerous for a cell. I've mentioned this before, because the cytoplasm is a solution, and it's a rich solution, and it's very often immersed in fluids which are less concentrated, more dilute. And so water tends to often flood into cells. And look what happened here. We increased the volume. Imagine that that's a cell, and we're increasing the volume. Well, there's a limit. Eventually, that cell can burst. And this is a very real danger for a living cell, that they can, in fact, burst like this. There are different circumstances that we can picture. This is actually a very easy little experiment to do in the lab, is to take red blood corpuscles, which is red blood corpuscles are living cells, and then put them into different solutions of different strengths. So in actual fact, in our blood, in circulating blood, our corpuscles are in a solution where the concentration outside, in the serum, is pretty much the same as it is inside the corpuscle. That we say is isotonic, like that, isotonic. This is the situation. The outside of a cell, that's a semi-permeable membrane. It's going to allow passage of water, but not the passage of salts. Water is going to pass in, and every molecule of water that passes in, one will pass out. Why? Because this is isotonic. There's an equilibrium here. There's a dynamic equilibrium. As one molecule moves in, one molecule of water will move out. And that's to keep the same concentration inside and out. It's a very healthy situation. And our cells are very happy at an isotonic, with isotonic body fluids around them. If I take the same red blood corpuscle and I put it into a hypotonic solution, that is one which is very much more dilute than the interior of the red blood corpuscle. Water is going to flood into the red blood corpuscle. It does it actually incredibly fast. Water just pours into the cell, and the cell will swell and swell until it bursts. A red blood corpuscle has no protection against bursting like this under osmotic pressure. Instead, it bursts or lyses. So this is a lysed cell. It's burst because of osmotic pressure that is built up inside it from the passage of water into the cell. The opposite situation, if I take the red blood corpuscle and I put it into a solution where the solutes on the outside are very much higher, then water is going to pass out of the red blood corpuscle to try and dilute the outside and to end up trying to make the two solutions equal. So, in that situation, the cell is going to lose water very rapidly, and it will end up shriveling like this.
Can you repeat that again, sir?
Yeah, sure. Where from?
From the hypertonic, yeah.
Okay, all right. A hypertonic solution is one where there's a lot more salts outside than inside the cell. So the outside is hypertonic to the inside. And when that happens, water will pass out of the cell. It will flood out of the cytoplasm. What is driving it is there's osmosis of water in this direction now. The water from inside the cell is passing out to the environment to try and dilute the solution outside. And as a result, the cell just shrivels up. It loses all its water from the cytoplasm and shrivels and will die. Eventually, the cell structure is lost and the cell can't survive. So those are three. Yes, go ahead.
If you add distilled water to blood cells, will the cells burst?
So show me on this. Which one do you think? This one, this one, or this one?
It's going to be hypotonic, right?
Hypotonic, yes. Distilled water would be extremely hypotonic, and the cells would burst. It's an amazing thing to watch. If you look through a microscope, you drop distilled water on them, you can just see the cells go and burst as the water floods in. So that's a very dangerous situation for cells. And there are many organisms which live in hypotonic environments. And when that is the case, they have to have some protection against bursting. And that is the function in plant cells and in bacteria and in fungi. That is the function of the cell wall. So the cell wall is something completely separate from the cell membrane, remember. The cell wall lies outside. It's not a barrier at all. It doesn't provide any sort of barrier to passage of substances. It's an inert structure around the outside. The cell membrane is on the inside. The easiest way to think about it is in plants. Plants have this inside the cytoplasm, they have this huge organelle here called the central vacuole. That's the principal controlling feature here. So this is a solution of salts. When you put a plant cell into a hypotonic solution, water floods into the central vacuole. It floods in to try and dilute the salts in here. Why? Because water concentration is higher out here than it is in there. Water moves in to try down its concentration gradient. When that happens, the cell swells. But now we've got a cell wall around the outside, and the cell membrane, instead of just going on to infinity and bursting, the cell membrane now stretches up, presses against that cell wall. And you can think of it, in fact, the cell in physics, terms of physics, the cell wall is exerting an equal opposite force on the cell membrane to the cell membrane pressing out. And the two equalize. and the cell doesn't swell anymore. But the cell now is under pressure. There's a pressure inside the cell, and in plants we call that turgor pressure. And the turgor pressure is very important for plants. This is the normal situation for plants. Plants, in general, exist in a hypotonic environment. The liquid around their cells is hypotonic to the inside. The cells swell and they have this pressure, and that pressure together with the cell walls is what helps a land plant to stand upright against the force of gravity. So it is the normal situation for plants that the inside of the cell is under pressure against the cell wall. If you take a plant cell and you put them into an isotonic solution where you've got as much water going in as going out, they in fact lose that internal pressure, and the plant will begin to look like it's about to wilt. If you do something really serious to the plant, like you don't water, you forget to water your house plants, right? This is what happens to them. If you forget to water them, then the liquids and everything around them start to become hypertonic to the cell. So they can become hypertonic. What is going to happen to a plant cell in a hypertonic solution? It's going to lose water. If you forget to water your plants, this is what happens to the cells. The cells begin to shrivel. First of all, they lose all their turgor pressure, and then they begin to shrivel away from the cell wall. And that ability to stand upright is lost, and the plant falls, it wilts, because it has no internal strength anymore. There's none of that turgor pressure to keep it upright. Water your plants, pretty soon they've moved to this situation. Water floods in, swells the cells again, and the plant stands upright if you haven't left your plant unwatered for too long. This is a special situation where the cell membrane pulls away from the cell wall like that. Is called plasmolysis. So this is a plasmolysed cell. Okay, oh yes, I just want to tell you one thing here. So obviously, red blood corpuscle is an animal cell, but a red blood corpuscle is typical of animal cells in that it has no cell wall. We have no cell walls. So, but our cells are subjected, and many animal cells are subjected to changes of environment, changes of tonicity. And in a hypotonic environment, animal cells are in a really dangerous position because of this. They're a danger of lysing. So animal cells will very often have some other method of regulating the passage of water. And this is called osmoregulation. They regulate the passage of water across their membranes. And for example, freshwater organisms have to have really specialized mechanisms for dealing with the water that floods into them the whole time. If we look at this simple ciliate called paramecium in fresh water, you see this here. That's a special organelle. In fact, they have two of them. This is called a contractile vacuole. And the contractile vacuole is a pump. Remember, this is existing in a hypotonic solution. This is in fresh water. Inside of the cell is very much more concentrated in solutes than the outside. Water is going to flood in the whole time. Now, the paramecium doesn't have a cell wall to swell up against to stop the passage, to regulate the passage of water. So instead, they have a pump. It's here, and it's called the contractile vacuole. And if you watch paramecium under a microscope, you will see this contractile vacuole fill and empty, fill and empty. And all it's doing is it's pumping out water, it's compensating, pumping out all the water that floods in because of osmosis. as I say, the paramecium actually has 2 contractile vacuoles. I think the other one's probably there. And you'll see one fill while the other one empties. They kind of pulse back and forth like this. And many animals which live in hypotonic environments like that, they have to have similar mechanisms.
Wait, so I have a question. I'm sorry about
Yes, go ahead. No, it's not bother at all. Go ahead.
Okay, so this thing that you're talking about, that allows water to enter and leave the cell, like that pumps out the water and brings in water?
It pumps out water. It doesn't have to worry about it. The water just floods in passively.
Okay.
Yeah, the water just floods in passively. I'll show you in a minute why that is the case. But yeah, the water just comes in. They can't stop the water coming in. So
Okay.
they've got to get rid of it somehow. Otherwise they'll burst.
Okay. So that's like the maintaining to like the cell, like that allows.
Correct. Yes.
Okay.
Yes. Exactly.
All right. Thank you.
Now we have an interesting situation. Okay. And this is to talk about fish, because fish are in an aquatic environment too. And they're in an especially dangerous situation because, for example, they have gills where all of the membranes of the gills are immersed in fluid so that they can pick up oxygen or whatever. So fish are at particular risk of changes in the osmotic nature of their environment. What we would say that in the tonicity of their environment, how much, whether they are hypertonic or hypotonic, is very important to fish, and they have to regulate very, very carefully. But I want to tell you that fish actually evolved in fresh water. They evolved in a hypotonic environment and then migrated to the oceans. And the ocean is not hypotonic, it is hypotonic. So now fish end up in a very interesting situation. How do they osmoregulate? Well, let's work it out. Just think about what the composition of fish cells is relative to their environment. They are in a hypotonic environment. They're in fresh water with a low concentration of salts outside, right? And instead, their cells are highly concentrated. Water is going to flood into their cells the whole time. It floods into their cells, especially through the skin, because the epidermis is exposed to the water, and then especially across the gills. Water just floods into their cells, and they have to deal with that somehow. And the way in which they deal with it is that they produce a very, very dilute urine. The main responsibility of their kidneys is actually to excrete water, get rid of all of the water that is continuously flooding into them. So that is how. Yes, go ahead.
They have, are there cells specialized, like have specialized vacuoles that also pump out the water, or no, it's only their kidneys?
No, it's much more that they have altered kidney function to do it. There are a couple of other mechanisms, but it's mainly the kidneys that do it. Yeah. So the freshwater fish, what they really need to do is they need to maintain a high concentration of ions inside their cells, and remembering that saltwater fish evolved from freshwater fish, we actually find that they're about the same. If we look at the tonicity of freshwater fish compared to saltwater fish, their interior is much more dilute compared to the outside. They have the same as freshwater fish. Now they've gone into the sea, which is a very high concentration of salts, 35 parts per thousand of salts, much higher. What is going to happen to, what is the direction of water passage from a saltwater fish? They lose water continuously. They lose water continuously to their environment. It just lost passively, especially through the skin and especially through the gills. And as a result, saltwater fish run a grave danger of dying from lack of water because they simply lose water the whole time. So what do? They drink. They drink water. They drink seawater continuously, and they then excrete the salt. They pull the water molecules away from the salt, and they excrete the salt, especially through the gills and the kidneys. Kidneys produce a very, very, very concentrated urine. It's a very concentrated solution of salt, originally derived from the seawater. Isn't that strange? The fish in seawater are in danger of dehydrating, of losing all of their water. And so they have to drink continuously. Yeah.
Why though?
Hi, what's up?
You said they're in danger of dehydration in the seawater?
Yes.
Why?
Yes. Because they're losing water continuously. Their interior of their bodies is much more dilute than the seawater. So water floods out of them. Water floods out, especially out of the gills, and the skin as well, but especially from the gills. They lose water to their environment the whole time. So instead, they have to drink water continuously, and then they take the salt out of the seawater, and they excrete the salt. They keep the water and they excrete the salt. They make a very concentrated urine full of salts, and they actually pump out ions from their gills as well.
Professor?
Yes, go ahead.
Aren't they losing nutrients though with the constant influx of water? Because you know how like when like humans overhydrate, sometimes they're losing a lot of like the electrolytes, and then they have to like drink Gatorade or something like that.
Okay, sure. We've only talked about the water, right? This is all physiologically balanced, so that sort of thing it does not happen. Okay, we're only talking about the osmotic balance, but for sure, all of this has to be run along with all of the rest of the physiology of the fish to make sure that they're not losing nutrients or whatever else. Yes. Okay, so here's, by the way, let's see if this works. I hope it does. This is a little video, and I described to you plasmolysis in plant cells. That is when the plant cell loses so much water that the cell shrinks and the cell membrane pulls away from the cell wall. So before I start, before I start, I'll just point out, this is a plant called Elodea. And it's a freshwater plant. So what they're going to do is they're going to put it into a saltwater solution. And the cells will begin to dehydrate as water floods out of the cell. Here's the cell wall there. This is a normal cell, right? This is all normal at the moment. There it is there. In the middle here, there's a central vacuole. And that's the organelle filled with an aqueous solution. Normally in plant cells, it is the central vacuole that is swelled and created turgor pressure against the cell wall. Now we're going to pour salt water onto this freshwater plant, and we watch. Look what's happening. Can you see? You see? Look there. Let's see it again. You see, that's the cell membrane there. Okay, so what is happening there is effectively that central vacuole is simply collapsing. All the water is leaving the central vacuole to try and dilute the salt outside. As that collapses, the volume of the cell just decreases and it pulls away from the cell wall. And that situation is reversible. As long as you don't leave them collapsed like that for too long, it is actually reversible. You can restore the cells if you do it quickly enough. Okay, so is everybody all right so far?
I have a quick question.
Yeah, go ahead.
So when we were just watching that video, it looked like the cells were like expanding. Is that when the water entered the cell?
No.
Oh, no.
Look at it again. No, they're collapsing. Okay, here's this normal cell. There's the normal cell there. Okay, and the cell is filling. This is the cell wall on the outside, that box-shaped thing there.
Okay.
So the cell is filling that completely. We've got to listen to her irritating voice. Look there, you see, the cell is collapsing.
Oh, I see it.
And it's pulling away from the cell wall. There's the cell wall. The cell is just collapsing completely. Okay. So, oh yes, you can, if I stop it there, you can see how the cells here have all collapsed and they've left the space. Here's the cell wall there. They leave the space here. And that's because the central vacuole has just collapsed. It's lost all its water to the outside.
Okay. That makes sense. Thank you.
Okay. All right, sure. So we've got to ask a question. Now, I said that water does not get across the membrane very easily. It doesn't pass that phospholipid bilayer very easily. So how is it that water just floods across the membrane like that? Well, the answer is that embedded in the membrane, there are special proteins which are responsible for the transport of substances across the membrane. There are various kinds of proteins, as we'll see. But this one here is called a channel protein. And a channel protein is one which facilitates diffusion of substances. So these proteins that we're going to discuss here both facilitate diffusion, and diffusion, in other words, they always operate from an area of high concentration to an area of low concentration. They obey that diffusion rule. And they do not require energy. They don't require an input of any biological energy to do their jobs. They do them in different ways. This one here, which we call a channel protein, it's embedded in the membrane. Okay, it's an intrinsic protein. It goes right the way across, and it has a channel that runs right the way through. And that channel is very selective. There are many of these channel proteins, and they allow very specific substances to cross them and get across to the other side, but it's always obeying the diffusion, high concentration to low concentration. They always down the concentration gradient. So if high concentration solute, whatever it is on this side, low concentration on that side, then the channel protein allows it to pass through. Channel proteins are very important because they are so selective. Some of them will only allow one particular ion to get across, for example. And there is one set of channel proteins like this, which are responsible for allowing the passage of water alone. They are specialized to allow water to flow, and they are called the aquaporins. So aqua water, obviously. Porin, because it acts like a pore. Remember it that way. They act like a pore to allow the water to flow. So there are many aquaporins in the membrane, and they allow the free passage of water. And it's they who really give the membrane its semi-permeable nature. They allow water to go across, but not other things. So that's a channel protein, easy to remember because it literally has a channel. This is another kind of protein involved with facilitated diffusion. Again, this is a diffusion process. It's going to go from high concentration to low concentration only. And it does not require an input of energy. It's a passive process. But this is called a carrier protein, and they operate a little bit differently, also very, very specific. But what happens is that they actually bind a molecule of what they are going to transport. This is how they acquire their tremendous specificity. They will only bind something that fits perfectly there. When that binding takes place, the molecule, the protein molecule, actually changes shape. And when it changes shape like this, it loses the ability to hold on to the solute, and it spits it out the other side. So here it has tremendous affinity for the solute, and it will actually bind it, change shape, and it loses its affinity for the solute, and it diffuses away to the interior like that. They can actually operate the other way as well. Yes.
Does this require ATP, or does
No.
the first one?
No, very important. Very important. No, this is a totally passive process. This does not require ATP. It is actually being driven by the diffusion pressure, if you want to think of it that way. It's being driven by the need of these atoms to pass, equalize down the concentration gradient, but it requires no biological energy. We'll see an ATP-driven one in a minute. Okay, I think, let's just see. I don't know how informative this is. Okay, so now that's all facilitated diffusion, obeys diffusive properties, and requires no biological energy. But very often, a cell will need to move molecules against the concentration gradient. That is to concentrate them inside the cytoplasm, for example, or inside an organelle, where they're already concentrated. And this process now, when you start to work against the concentration gradient, is going to require energy, and it's going to require biological energy. And these are the systems that we know as active transport systems. Again, these are protein-driven systems. So, for example, this here is a very important one called the sodium-potassium pump system. And this pumping system actually operates especially importantly in nerves, because we have to concentrate to get nerves to function. I won't go into detail now, but to get nerves to function, you have to concentrate sodium in high concentration on one side, potassium in high concentration on the other side. And this is called the sodium-potassium pump. And if we just the inside here being the cytoplasm, it will bind three ions of sodium. When it does so, they're going to sit there. They actually change the nature of the protein and make it interact with ATP. And this protein becomes phosphorylated. It takes a phosphate from ATP, and that provides energy and causes the shape of the protein to change. And it now spits out its sodium to the outside, where there's already sodium. So this is moving sodium into an area where there's already high concentrations of sodium. This protein can also pick up phosphorus. And so here we see here, as they bind here to the protein, and now that phosphate bond there is broken, and the energy is harvested to change the shape of the protein again and spit them into the inside. So this sodium potassium pump actually works to move the two ions in opposite directions and accumulate potassium on the inside, accumulate sodium on the outside. This is for a particular reason. Eventually, we're going to allow these different ions to flow, and we'll create a little effectively a little electrical nerve impulse by doing that. But to get it to set the nerve up, we have to accumulate sodium on the outside and potassium on the inside, and it's done by the sodium potassium pump. This is an active transport system. It is deriving energy from ATP, first by phosphorylating, by adding a phosphate to this protein, and then by breaking that phosphate bond and harvesting the energy to cause the movement of the potassium in. Is that okay? All right. Let's see. Okay, so that's there for you to revise. This is just a summary slide for you to emphasize the difference between the passive transport systems, the ordinary diffusion, just straight across the membrane, a couple of things, gases, things like that will diffuse across. But then most other substances, their movement across the membrane has to be facilitated, and it's facilitated by proteins. In this case, this is a passive. This is facilitated diffusion. It is a passive process, either through a channel protein or through a carrier protein. The active transport systems are always going to use biological energy, and they do so because these systems require an input of energy to move substances against their concentration gradients, to move them from an area of low concentration to an area which is already high, to store them and to accumulate them to some purpose. You just realize something. If I accumulate, let me just say this. If I accumulate sodium ions on one side of my membrane here, I've done two things. I've set up a chemical gradient of sodium, right? I've got more sodium on this side than on that side. But I've also set up a charge differential. I've got more sodium positive charges on this side than I have on that side. So I have two driving forces which can try to push those ions across this membrane. One of them is need for chemical diffusion. The other is to equalize the charge. And we call that an electrochemical gradient. And I've used this analogy with you before. The cell will often do this kind of thing. Often, for example, we will accumulate protons on one side of a membrane relative to the other side of the membrane. It's the same thing. It's an electrochemical gradient because you've got more protons, hydrogen, on one side than on the other side, but we've also got more positive charges on one side than the other side. And if I've got a barrier, an insulator, that's what this membrane is. It's an insulator. If I have an insulator and lots of charge on one side and not on the other side, what have I got? That is a battery. It's a system which sets up potential energy, tremendous amount of potential energy. And if I can let that electrochemical gradient equalize, I can harvest that potential energy and I can actually use it. And the cell very often does this. We'll see it in all sorts of different instances later on. But just remember, the cell, if you're wondering why does the cell accumulate things on the outside or on the other side of a membrane, that is one of the important reasons why it sets up a situation of potential energy which the cell can harvest to do something useful.
I have a question.
Yes, go ahead.
So in an active transport, can you explain to me exactly what an ATP is?
Sure. We haven't discussed it in any great detail. ATP is actually a nucleotide, adenosine triphosphate. And it has, so it's triphosphate, it has it's phosphates attached to it, 3 phosphate groups. It's that first phosphate group it's the third one to be added. It's that one which has the most energy associated with its bond. So you can picture, we've got ATP, and then we've got 3 phosphates, 1, 2, 3. This third phosphate there, that phosphate bond has a lot of energy attached to it. And if we break that bond, we can harvest the energy. And that is the routine coinage of energy in biological systems, is to use ATP as a source of useful energy. And later on, we're going to talk about how ATP is made and how it functions in the cell. But for the moment, you can just picture it as being, it's not long-lived, but it's a temporary store of energy which the cell can access to use. And the system that we talked about here, where it phosphorylated this protein, that is a very frequent way for the cell to use the energy in the phosphate. It's harvesting energy in that phosphate bond to do work in the cell. For the moment, just remember, ATP is a nucleotide and it has 3 phosphates attached to it.
Okay,
and
thank you.
It's usually the first phosphate. We can take it down one more, but it's usually only the first one that is used.
And that's what makes it an active transport, right?
Correct. Yes, it's active transport because we actually require energy to drive the process. Otherwise, it won't work. It requires energy to be activated.
Okay, thank you.
Sure.
Professor.
Yes.
What helped me remember was that since it's active, in order to do an activity, you need energy. So that's how I remember that it requires ATP.
Yes, that's a good way to remember. Yeah, that's a good way to remember. And remember, we always phrase this as saying, this is biologically useful energy, because the cell is full of energy, but the cell can't necessarily access it. It can access the energy in ATP. ATP is biologically useful energy. Okay, so there's just one other kind of transport that we won't spend a lot of time discussing, but I just want to mention it. And this is called a co-transport system. They're actually quite common in the cell. And what happens, it's an active transport system. We're going to see that what happens is there's one part of the system which requires an input of energy. This is a very common way for this to work. For example, this is a proton pump. A proton is a hydrogen ion. it's a hydrogen nucleus which has lost its electron. So it's just a single positive charge. That is all. So this is the proton pumps are very important because they pump the protons out to one side of the membrane. And here we see how we've accumulated both a chemical difference and a charge difference. We've got masses of positive charge on this side relative to that side. That is an active process. It requires an input of energy from ATP. What has happened here? Well, we've set up a system where we have a lot of potential energy. The drive actually is for this gradient to be equalized. These protons can't pass across the membrane, however. They can be accumulated on this side. The drive is both a chemical drive and an electrical drive because we've accumulated charge here. So we've got more charge, positive charge on that side. This side is relatively more negative to that side. We've also accumulated a chemical gradient. We've got hydrogen, protons on this side and not on that side. I'll just remind you, if we accumulate protons in a solution, in an aqueous solution, we're making it more acid. So this is more acid on this side. You can think of it that way as well. So we've got here a lot of potential energy locked into these protons sitting on that side of the membrane, and we can harvest it. We see this in many different systems. We'll see this in respiration, for example, but in this system here, it's being used to transport substances across the membrane. And in this case, it's called a sucrose transporter. Here's sucrose, that's table sugar. And sucrose consists of a glucose and a fructose joined together. And the sucrose molecule, it's actually quite difficult to get it across the membrane. So what we do is what the cell does is it has dedicated proteins which will only transport sucrose. And the way in which they do it is they allow a proton, they bind a proton and transport it across. And they're actually able to harvest the potential energy. This proton is moving down its concentration gradient, and it actually releases energy, and that energy is used to move the sucrose across on the co-transporter as well. So it is an active system, although this is the only place where we actually utilize ATP. But we build up potential energy on that side, and we harvest that potential energy by allowing the protons to flow, and at the same time, we use that energy to transport sucrose. So that is a co-transport system, and this is a proton sucrose co-transporter. They're usually named for the substances that they will transport. There are others in the cell as well. Okay, so all of those are what we were talking about there. What we've been talking about are relatively small molecules. Okay, so they're single molecules moving down their diffusion gradients, but there is also a case that the cell may sometimes need to move very large molecules, and in both directions. Move really large molecules out of the cell and take very large molecules in, where those kind of transporter systems don't work. Instead, the cell takes in molecules like that, takes in very big molecules in bulk. And that is why this is called bulk transport. These are what we're going to talk about now, are bulk transport systems. And I'll show you the first one is quite easy to understand. And this is a process called exocytosis. And this is typical of any cell, for example, which is excreting something. Maybe it's a glandular cell that's producing a substance. Maybe it's an immune cell, which is releasing antibodies, for example, something like this. Remember back to our discussion of cell function. And you'll remember that we said substances are transported around the inside of the cell in vesicles. These are spherical organelles containing and isolating a substance. And the one place where these are very often made is on the Golgi apparatus. The Golgi apparatus modifies substances, largely proteins, but polysaccharides as well, and various other things. But the Golgi is a site of secretion. It's the site where the substances are finalized, that the cell is going to excrete, whatever they are. And what the Golgi does is it will wrap those substances in a vesicle, in membrane-bound vesicle. The membrane, by the way, very, very similar, don't forget, to the cell membrane, to the outside, very similar to the membrane of the organelles. And when the vesicle is made, it is sent to a membrane, and because it's so similar, it's actually able to fuse. It is able to fuse with the membrane, and you see that what it's done is it's opened itself up to the outside, and it releases the substance that it was going to make. That is exocytosis. That is the cell spitting out. Sometimes this can also be used in cells to get rid of waste material, but most often see it as the cell produces products that it is dedicated to being, like a glandular cell or something like that. We would see a lot of exocytosis of those substances. So I'm going to return to this diagram in a while just to tell you one thing more. So we also have a situation where the cell may want to take in substances from the outside. And there are three circumstances that we recognize. The first is one which we see frequently in single-celled organisms, for example, like amoeba, those sorts of things, which feed from the outside. They actually they extend part of their cell membrane out and catch things and enclose them. And they enclose them by this process called phagocytosis. And that literally means feeding the cell. So what they do is they extend out of pseudopodium like this. They wrap it all around, and then they clip that off to make this. That is a food vacuole. And this is a typical example of endocytosis, taking into the cell from outside. And it's specifically here called phagocytosis because it's feeding. It's a feeding response. So phagocytosis is an endocytosis.
This happens in an animal cell?
This happens, you will see it in animal cells. For instance, you might even see it in intestinal cells and things as well. But in things like protozoa, protists, free-living, single-celled organisms, it's the commonest way for them to feed, is by phagocytosis.
Because plant cells, they don't have food vacuoles, right? They only have water vacuoles.
Correct, correct. They can't have this, they don't have anything like this because they have a cell wall which prevents them from being able to form this kind of vacuole. So I just remind you of your cell discussions earlier. You'll remember that food vacuoles very often now will unite with another organelle called the lysosome, which contains digestive materials, digestive enzymes, which will break down the food in the food vacuole for it to be absorbed. So that is phagocytosis, which is an endocytosis. Here is another type of endocytosis. This is called pinocytosis. And the pino refers to the fact that it's very small. And that is what's characteristic about pinocytosis. It's sometimes just called cell sipping. They take very small amounts from the outside into the cell. And I tell you that this is especially important in the immune system. There are cells which continually sip the environment to check to see whether there are viruses or toxins or anything in the environment. I also mentioned, actually, we were talking to animal cells, our white blood cells in blood circulation. They show very vigorous phagocytosis. They will eat bacteria, for example, very vigorously. So this is pinocytosis. It works through a special system whereby the membrane is kind of buckled. A special set of proteins is laid down that cause it to buckle. Don't worry about it. It just forms a very small vesicle containing a mixture of substances from the outside, often for it to be analyzed in some way or another, as the immune system would do. So here's the last kind of endocytosis, and this is called receptor-mediated endocytosis. And this occurs in response to the presence in the environment of specific substances. And the proteins embedded in the membrane here are actually sensory, as well as causing various things to happen. So for example, in a receptor-mediated endocytosis, we have these proteins here embedded in the membrane, which bind specifically to a particular substance. Very often, these occur within proximity to one another. And if enough of them bind the substance, they will send a signal to the cell that causes the cell membrane to buckle in this fashion and to cause a vesicle to form. And you'll see what's happened. Here are our proteins embedded in the membrane, right there. They bind the substance that the cell is interested in. And once enough of them have bound it, they cause this vesicle to form. And look what's happened. The vesicle has formed with the substance still bound to the proteins on the inside, like that. Now the cell can proceed. Now it can proceed to digest this, for example, or make use of it. We use these receptor-mediated endocytosis systems for a variety of purposes. Sometimes to accumulate nutrient. Other times it's a response to presence of hormones or this sort of thing.
Professor?
Yeah, go ahead.
Once the food travels like in the food vacuole, goes inside of the cell, does it get broken down into energy?
Yes. In this case here, you will have a lysosome merging with this food vacuole to digest the food. I just want to emphasize this is very particular for food vacuoles, for taking in food. These pinocytosis and receptor-mediated endocytosis, there are a wide variety of reasons why the cell will do this. It's usually not for feeding. It's usually to accumulate information from the outside. It's often, in this case here, often a response to hormones binding here, for example, or that sort of thing. So once the vesicles are made, then they are processed appropriately for whatever purpose that system exists. These will be, just remember, this is really large. Pinocytosis, these are small. Receptor-mediated endocytosis, these are also usually the vesicles are quite small. Okay, I think we just got some pictures. This is an amoeba eating an algal cell. But remember, our white blood cells, especially macrophages and neutrophils, both feed in exactly the same manner as this, forming a food vacuole that is then digested. This is pinocytosis, and you can see here the pit forming, and it's small. Here it is here, taking in substances from the outside. And the proteins that make it here are called coat proteins. And when they form like this, they just mechanically, doesn't require energy. They mechanically just form a basket that pulls the membrane in that fashion. So it's called a coated pit. And this is a coated vesicle, coat proteins around it. And again, this looks quite similar, excepting that this formation of this coated vesicle is in response to these receptor proteins binding something in their environment. And remember, these are very, very specific. They're very specific for one particular molecule that they will bind. Okay, so that's it for today. I just wanted to say one thing about the about these recorded classes, about the classes in general, and when you use the recordings. Obviously, if you're sitting watching this live, you can't do this. But when you go over these as recordings, I strongly advise you to break them up. It's like 20-minute segments and stop after 20 minutes. We've gone like an hour and 10 minutes, and I know that your brains are fried. And that is completely natural, right? So when you're revising, break these into segments and stop the video, and then go back over the notes, go back over the PowerPoint printout, check the slides, make sure you understand the slides. If need be, rewind and listen again, and take a break. Take a minute, 2 minutes, just to do something completely different, and then sit down and carry on. Give your brain a break every now and again. About 20, 25 minutes is actually the most that any human being can carry on concentrating on one thing, like a lecture or whatever else. So be kind to yourselves in your study. Again, this will be put up as a recording. And now, if you have any questions, fire away.
I just have a question about the homework. So you give it every week, like the 50-part thing?
Yes, unless there was one that went over 2 weeks. But then you'll get it. You know, I usually I try to put them up on Wednesday afternoon, but sometimes I only get them up on Thursday. But usually Wednesday or Thursday, one will go up. If it's very long, then I'll set it to go for 2 weeks, which I think one recent one was for 2 weeks.
And we have a quiz every Wednesday, so we won't have one.
Oh yes, thank you for reminding me.
No, I didn't mean to. I was just asking.
Yeah, no, I did mean to tell you. Yes, there will be a short quiz. There will be a short quiz on a Wednesday at the start of class. Last time I set it at 1 o'clock. I'm very sorry. I should just set it at the start of class time, 1:55 on Wednesday, and it'll be for 10 minutes, and you can probably finish it in 5, I would think.
I'm gonna need that 10 minutes.
You did take 10 minutes?
Yeah, I'm gonna need more than that, a little bit.
Okay, all right. I can do that. Yeah, well, I gave you 5 questions, right? So yeah, we can set it for more. We can make it for 15 minutes.
Thank you.
Thank you.
Yeah, Professor, I could definitely use that extra time, because I have to reread the question and to just go over it, since there's such a large amount of material that we're required to remember and learn. I just sometimes I like to focus on all the choices and really think what makes sense.
Okay.
Yeah.
All right, but don't forget my advice about multiple choice questions. Always try to have an answer in your head before you go through sometimes the question is you can't do it because the question is structured, so you have to read all the answers first. But if it's very often a multiple choice, you read the question and an answer should occur to you, and then look for your answer in the list.
Professor, I have a question.
Yeah.
Sorry.
No, go ahead.
So the test that we had last Wednesday, I think it was, you put it for as you said, for 1 o'clock instead of the start of class time. And yes. I didn't know about that, so I didn't take the quiz.
It's all right. Don't worry. First of all, missing a quiz is not going to make any difference to your grade. But I think what I'm going to do, I'm just going to credit everybody because several people made a mistake on the time. And then this week we will make it definitely at 1:55 at the
Okay.
class time. All right.
Thank you.
Okay.
I have a question.
Yes, go ahead.
I start work this week at 1, so I won't be able to attend the quiz. Is it possible to have an extension?
For the quiz,
'Cause I'm
yes.
gonna be at work.
Yes, that's all right. Yeah,
Thank
yeah,
you.
for the quiz, yes, not for tests. So, yeah, you're gonna have to make some arrangements for the test, because the test I want everybody writing at the same time.
Professor, do we have a test coming up?
No,
We have
I
an
forget.
exam.
When is it?
An exam on like the 27th, I think.
Yeah, because that's the
The 26th.
26th. Oh, excuse me. 26th.
okay, all right, yeah. And that, by the way, I think is what counts as the midterm, but it's not it's a test like any other test. It's on stuff that you haven't been tested on. So it's the stuff from the end of the last test up to wherever we get then. Not on everything from the semester.
I have a question.
Go ahead.
For the Wednesday quiz, so it's only 6 questions max
Yep.
for 15 minutes, or
Yes, it'll be for 15 minutes.
okay, thank you.
All right. Anyone else?
Professor, you're also going to be hosting like your all open office hours.
Oh, yes, on Thursday. Yes, yes. I'll send out an email to remind everybody.
Thank you.
Okay. All right, everyone. Nice. Well, I don't actually see you that well. It's nice to know you're out there. Put it that way. Okay.
All right, bye.
Bye-bye.
Thank you so much, Professor. Take care.
Thank you.
Yeah, you too.
Have a good day.
Thank you.